We'll move to the last but not least presentation by Brian Corrie. So Brian Corrie is a computer scientist, project leader on the iReceptor and iReceptor Plus project. So, and he's going to talk about the AIRR Data Commons and how to make AIRR-seq data fair. Thank you.
Great, thank you. And Cornita, you can hear me okay?
Perfect.
Great. Yeah, and Christian did a great introduction to kind of what I'm going to be talking about. I'm going to focus more on how you can practically use the AIRR Data Commons, which is this network of AIRR-seq data repositories that Christian was talking about. And Christian also covered a lot of the background, and a lot of the other background that I'm going to talk about has been covered already. So I think I'm not going to spend too much time on my first couple of slides. I think the big ticket item from this slide is that the adaptiveness of the immune system is really the key thing that makes it so hard to understand, and that's what we're struggling with in dealing with these types of data. And I think a number of people have already talked about this, but I'll just go over one more time just to highlight it. So it's estimated, these are all estimates, I don't think we really know, but it's estimated there are about 10 to the 13 possible immune receptors in the human population, and any individual at any given time can have approximately 10 to the 8 cells per person, and that's at any given time point, and over time they change. And typically when you do AIRR-seq data sets and AIRR-seq sequences and studies, you're sampling that 10 to the 8 sequences or cells, sorry, with probably at most you're getting some sequences or some samples that are sequenced with about 10 to the 6. So you're at least 2 orders of magnitudes a statistical sampling of the population, and so that's a big challenge that we face. And I think a lot of people have alluded to this, but it hasn't been really stressed too much. But there's a very, very large number of studies that are being carried out, and each one of those studies is very complex. So you can have many subjects in a study. You have multiple time points. Nina was talking about her study where she had pre-vaccination, post-vaccination, pre-second vaccination, post-second vaccination. So these are complex studies that are taking on the order of 1 million sequences per sample, per time point, across many subjects. You have many studies that are doing this kind of work in various labs around the world, and you've got hundreds of labs around the world. So these are really, really large data sets. They're complex data sets. And I think one of the things that kind of Christian pointed out is that, imagine if this data was available to you on the internet, as Christian said, right? So you don't have to sequence a set of healthy controls. There are a bunch of healthy controls out there that you can use. I think Victor actually raised a really interesting point about how can you tell whether those healthy controls are applicable for what you need them for. So that's a really complex problem. So Christian talked about FAIR and introduced that concept for me, which is great. So thank you for that, Christian. I won't spend too much time about it. But I think I'll drill down a little bit into how researchers use FAIR AIRR-seq data, or they could use FAIR AIRR-seq data. So the first part of the FAIR acronym is, of course, people need to be able to find data of interest. And this is about what, as Christian was saying, about being able to look at or find some data that is out there that you can use in your studies so that you don't have to reinvent the wheel over and over and over again. Many people have done many, many studies over time. There's a lot of data out there. How do you find it, right? Once you find it, it has to be accessible. Again, Christian mentioned this. So you have to be able to get it and access it and be able to download it and use it, okay? And for you to use it effectively, it has to be interoperable. So this is where the standards come in, and I'm going to talk about this in a little bit more detail. But ultimately, the goal is to be able to reuse that data. So we want to be able to empower users that are doing this type of research to be able to reuse that federated data in new analyses to derive new insights. So these new insights are driven by the reuse of that federated data. So what is the AIRR Data Commons? So as Christian mentioned, it's an international network of distributed repositories, and those repositories are distributed around the world. Currently, the AIRR Data Commons consists of 4 repositories, contains data from 60 studies, and from those 60 studies, there are over 4 billion sequences. It's important to point out, and I'll kind of reiterate this a little bit later in my talk, what those sequences are. So these are not sequences that are raw sequences. They're just the nucleotide sequence that you get from the sequencing machine, but these are curated and annotated sequences. So they're, the way I think of it is they're valuable sequences. So they've been processed, they're available to use without the researcher having to do a lot of research about what the study was about, how the study was performed, how that data was annotated. So one of the key things from a FAIR perspective is that these repositories are based on standards, and these standards are the standards that are developed by the AIRR community. Christian talked about these already as well, as have another other people. They have emerged from the AIRR working groups. There is the MiAIRR standard for study reporting. There are the file formats standards for data sharing. And there is the ADC Web API standard for querying data repositories. And the AIRR Data Commons Web API is the key one from an AIRR Data Commons perspective as to what the key is, because the goal is for the researcher to be able to query a repository and query that repository or a set of repositories around the world that are basically using the same language for that query. So you send the same query to a bunch of different repositories and you get the data back. So one of the key things is we want to make this easy. So one of the things that our project is working on is the iReceptor Scientific Gateway. So the iReceptor Scientific Gateway is basically a web API or a web-based platform for doing data discovery and analytics. And it basically queries the AIRR Data Commons. So it uses the AIRR Data Commons Web API to query the AIRR Data Commons. Basically, it tries to hide the complexity of the fact that you're searching tens or possibly hundreds of repositories and finding data across those repositories. So essentially what the user does is they send a query. The query goes out to the repositories, and the web portal federates that data and gives you a response back. So essentially what the user is doing in this case is basically sitting in their web browser, typing a query saying, I want all of the AIRR-seq cancer studies. I want a specific type of cancer. I'm interested in female patients that are over 40 years of age. You basically type those queries into your web browser. You click a button. That query gets sent out to 10 different repositories. You get some responses back with the data that you're interested in. So what is available in the AIRR Data Commons? As I mentioned earlier, there are 60 studies currently available, over 4 billion curated annotated sequences, and I'm going to talk a little bit more about that curated statement. So again, these data sets have been curated by typically a data curation expert. They've been annotated with all of the metadata from the MiAIRR standards that are possible to extract. They're annotated with the VDJ annotations. They've been run through an annotation tool. They've been run through a quality control pipeline. So these data are not the raw sequences. They're definitely highly curated with a lot of added value. So the AIRR Data Commons has been growing quite rapidly. You can see in the graph here, it goes from kind of early 2000, mid 2017 to 2021, basically the last couple of months. One of the things I'd like to point out is that over the last year or so, there's been an enormous amount of activity around COVID-19 and COVID-19 data. So currently we have 15 COVID-19 studies available in the AIRR Data Commons with over 1 billion annotated sequences. And this data is moving really, really fast. And one of the the interesting things about, I think, the power of the AIRR Data Commons and the potential that it has in terms of data sharing is that we've seen basically data reuse grow at a pretty substantial pace. So we had early studies published in June of 2020. Data was online shortly thereafter. And as you can see down at the bottom, in August of 2020, one of the really important studies, that was heavily reused, was published in August. And we had papers published that reused the data from the AIRR Data Commons in September, January, and April. So people were able to reuse this data, turn it around, and use that data in their study and get and have impacts on their results in later work. So how do you use the AIRR Data Commons? Again, one of the easy ways to do that is through the iReceptor gateway. You're able to search these 60 studies using the 80 MiAIRR subject sample metadata fields. I think, Christian, you said 63. I thought it was more like 80, but you're probably correct on that. I'll just use a quick example. So there's over 4 billion sequences in 60 studies. It's as simple as typing COVID-19 into a text search field, and that immediately gets you down to the 1 billion sequences of COVID-19 data that are currently exist in the AIRR Data Commons. So this search is actually sending queries to each of those repositories. So there are currently 4 active repositories in the AIRR Data Commons. So this query is being sent out to those repositories, and the data and the query response is being federated by the gateway to allow you to access that data. And at this point, you can kind of do some further analyses. You can look at vgene usage, dgene usage, junction length, histograms, so some simple analyses. And of course, you can also download the data for further detailed analysis. One of the things I wanted to point out, just to kind of inspire usage of the AIRR Data Commons, so we have seen a dramatic increase in new users on the iReceptor platform. This is my best way of kind of gauging use of the AIRR Data Commons. And in particular, this really large spike in new users between June and July of 2020 is when the COVID-19 data became available in the iReceptor platform, or in the AIRR Data Commons, sorry. And so kind of previous to that, we would have maybe 10 new users added to the platform per month. When the COVID-19 data was announced, we actually had, as you can see from the graph, over 70 new users apply for accounts on the iReceptor platform. And since then, we've had between 20 and 30 new users ask for accounts per month. One other thing that's worth pointing out is the usage of the AIRR Data Commons is quite broad. So we have international users, very large number of users from North America, Europe, and Asia. Another interesting thing to point out is there's a significant number of commercial and government users as well. So this yellow slice over here, this 52% or 18% and 52 users between June and March of the last year. So it's a very active and international platform. So one of the things I want to kind of stress is sharing data. So we have a lot of data in the AIRR Data Commons currently. It's been heavily utilized, but really this presentation is a call to participate. So we encourage everyone to share their data. The value of the data is only as valuable as the amount of data that we have in the AIRR Data Commons. We've spent a fair bit of time over the last several years curating the data that we have available, the 4 repositories that are currently active. But really, the way to make this a really, really critical resource is having more and more people share their data. So this is really a call for sharing. And there's 2 real key mechanisms to do that. Of course, you want to use a repository that implements the AIRR Data Standards. There's two mechanisms for doing that. You can run your own repository. So our project, the iReceptor Turnkey, has developed a software stack for sharing AIRR-seq data. So it implements all of the AIRR standards. It's easy to download and install, and I'd encourage you to have a look at that if you're interested. If you're a research group that produces your own AIRR-seq data, please have a look at that platform. And the other platform is the VDJServer platform. That's Lindsay Cowell's group. So Lindsay talked earlier today. And the VDJServer platform will actually allow you to publish your own data through their platform. So you upload your data, run it through the VDJServer pipeline and publish it, and that makes it available to the AIRR Data Commons and the broader community. So my takeaway messages from this talk. So the AIRR Data Commons makes AIRR-seq data fair, and there is a lot of data in the AIRR Data Commons today. But it is only a fraction of the data that is out there. I think the more data we have, the more value in the AIRR Data Commons. So please consider this a call for sharing your AIRR-seq data. Reach out to your colleagues in the AIRR community. There's a lot of people that can help if you're interested in sharing your data. And with that, I will wrap up. I'd like to thank my colleagues in the AIRR community. Christian had a very nice list of all of the different working groups and the timeframes. The AIRR community has been working for 5 years. It's a very dynamic group. We would welcome more people to contribute to the AIRR communities. People are welcome to join all the working groups. I'd also like to thank our collaborators in the various funded projects before. And I'll stop there and open it up for questions. I'll just leave it with a couple of useful references for people with some links at the end.
Thank you very much, Brian. Corrie, you have a question.
Hey. No, I don't have a question for Brian
yet.
Yeah.
So actually, I had a question on the sharing. So one important issue when we are dealing with human data is the protection of the data. So what can you say about this, and what are the rules so far?
Yeah, so currently in the AIRR Data Commons, all of the data is public data that has been published typically at SRA or ENA. And so all of the data is public. We recognize clearly that that's, data curators need to protect their data. The iReceptor Plus project is working on a platform for providing secure data. And so the iReceptor platform is, Christian mentioned this European Canadian consortium. A number of the people that have been talking at this session are part of that project. And we're working on a security platform for managing protected data. And so that's something that you can expect coming down the pipe probably in the next 6 to 8 months, I think. And so that will allow data curators to protect studies and share it only with those people that can authenticate against a repository.
Brian, can I ask a follow-up on that? So who will manage that authentication step? Those would be shared with MTAs and whatnot, or?
Yeah, the local curator and the local data steward would manage their own repository and manage access control to the data in the repository to the individuals that they had MTAs with.
I guess in a way we would, I was going to say, I guess in a way we would say those data aren't actually in the AIRR Data Commons then, though, right?
Yes. Yeah. So I mean, the goal with the security layer is to make it easy for a researcher to kind of live in both worlds, and recognizing that when people publish their data, that they can make it public at that point, but it's very, very valuable to them to have that data in a repository while it's private data as well. And so it's, you know, the goal for kind of security layers is to manage the transition from when you need to protect data and when you can share data. And of course, if the data is public, it's in the AIRR Data Commons. If it is not, if it's protected, then it's not technically in the AIRR Data Commons, but the goal is to make that transition as smooth as possible.
Can I ask one more question? Very nice talk, Brian. One question that I have is, so once the data goes into iReceptor and all these repositories, then it's pre-processed, right? And of course, these pre-processing pipelines evolve over time. So what do you think about, you know, being able to have different versions of the data in the future, pre-processed data, so that we can compare how these pre-processing pipelines, you know, maybe change the data, maybe also, since, as we have seen, very small differences could make the difference between, you know, health or disease. So how do you see that? Yeah.
And so the AIRR standard has mechanisms for gathering metadata about that. So you can annotate with metadata saying that this data set, these sequences have been processed with this pipeline. These sequences from the same study have been processed with another pipeline. I think when you go down that path, the danger is, I think, and I think, Victor, you asked the question, is how can you tell when those two situations are occurring, right? And so that makes the ecosystem more complex, because you've got the same data set annotated with different annotation tools and different annotation pipelines. And so, yeah, it's a very complicated space. And I think the critical thing is, and Christian kind of alluded to this, is we have to get the standards right. We have to get the metadata annotation right. People need to be able to tell when it's appropriate for them to use a data set and when it's appropriate for them not to, right? Because not all data sets are comparable, and the same data set may not be comparable if it was processed with different pipelines. And so those are all things that I think that the really important thing, I think, is that people need to really carefully think about when they curate a study, that they manage the metadata properly so that they can actually, so people can reuse it, I guess, correctly or validly from a scientific perspective.
Thank you.
I think it might also be worth commenting, again, for people who may not know this, that there's also sort of the data in a tin can that you put into SRA, right? Where you have the raw data, so that if you need to, you can go back and I mean, this is a pain in the neck, of course, but you can go back and reprocess the data with newer techniques if you need all the data to be kind of more uniformly processed after your favorite new tool comes out, Victor.
I mean, one of the things, I mean, ideally you'd be able to get to a place where you could look at the metadata in an AIRR Data Commons repository, go to the SRA, pull the SRA data out, run a pipeline on it, and get exactly the same answer that's in the AIRR Data Commons. I mean, I think that's the, you know, would be the ultimate goal. So then if you did want to go back and do something else, you could. You have a mechanism to reproduce the science, and you get a lot of really nice outcomes when you can get there. We're not there yet, but that's kind of the vision, I think.
That would be perfect, yeah.
Thank you very much. Thank you, everyone, for your great talk and the discussion. So we are running late, but there is 4 questions in the chat, 3 for Corrie and 1 for Christian. So I promise that we will address those questions or provide the answers. So let's go for them. 1 question. So Corrie, do investigators take the germline variation into account when they report number of somatic hypermutation? And if not, how much a problem is this?
That's a complicated question. Sometimes it definitely is an issue if you don't. But I think, you know, that's something that this community has been discussing for a long time, and we don't have perfect solutions for. But there is definitely a recognition that, particularly in humans, a lot of us have carried alleles that aren't currently documented in these germline databases. And so that's an example of when, if you don't account for that, you might get slightly misleading results from your somatic representation analysis. But there are pipelines out there that one can use to try to get around that, to sort of create individualized germline databases. And I think these are sort of now being used more often than they have in the past. But yeah, I think it's complicated. The goal is to sort of be improving the databases as we go, and individuals doing that in their analysis and sort of being aware of those things. But it is a bit of a heavy lift for new groups to get all of that right. Even admittedly, our group doesn't always do what we should, because it can be quite complicated when you have hundreds of samples to get individualized germline databases for all of them. But I think that is the goal ultimately.
So kind of related question from Maria Gutierrez Arcelus. She's asking whether there is associations between IG gene usage or germline variants and diseases, and if so, which ones?
Yeah, so there are. There's admittedly not a lot, and there's lots of reasons that I could talk for a very long time and bore everyone to tears as to why I think that is. But there are some examples where, efforts like GWAS studies have uncovered variants that are associated with disease. One of the most recent ones is rheumatic heart disease, and so which I think will be replicated, is in the process of being replicated. So I do think that there are these associations there. But again, the technical issues around doing these genetic association studies in these regions has really probably limited our ability to ask that question effectively. But there are a handful. Alzheimer's disease is another one. But the biology behind this hasn't really been investigated in any great detail. So even in these cases where we have seen these associations, it's not really that clear what's driving the sort of disease from these regions. Yeah, and then there's these other examples where we know that these, specific germline alleles contribute to things like vaccine response or response to specific antigens like flu and whatnot. But I think, the more we kind of catalog, the more we understand, the more we'll have the ability to ask those questions in a more direct way and get the answers head on.
Yes, and definitely the potential link of these variants with the generation of the diversity, the recombination process, that would be really interesting. But of course, we will need a lot of data to come.
Yeah, right. And that's one of the motivations for what we're doing, right, is really trying to make these first order connections, repertoire, and the genome. You can kind of define how that's working. Then the hope is that there's things that can be layered on in the context of disease or vaccine response or whatever it may be that you're interested in. I mean, that's not that different than what has been done elsewhere in the genome for other parts of the transcriptome. We try to understand these connections between genetic variants and then try to link that back to disease. So it's not like really a new model, it's just kind of a new model for this particular space.
So the last question for you, Corrie, is do you expect germline TCR variation may co-segregate with HLA haplotypes at the population level? And has anyone enough data to look yet? From Simon Malal.
I hope there, because I think that would be a really cool biological story. So I hope that there is there, because that's there. I don't think anyone's really given that a fair look. Mainly again because folks haven't really paid too much attention to TCR variation. I mean, there's a few papers out there that have. There are a couple papers in literature looking explicitly at how does HLA influence repertoire. Victor mentioned some of this work earlier on today. But I do think that there's a possibility there for it, for sure. And I suspect that the kind of evolutionary trajectories of the TCR loci will look different than what you see in IG, precisely because there is this kind of codependency with HLA. But yeah, I think we're nowhere close to even understanding what that might look like.
Yes.
But I think it's a really great
TCR
question.
people.
Yeah.
TCR people are lazy.
No, we're all lazy. No, we're not lazy. It's just, you know, you work on what comes in front of you. And I don't think it's come in front of anyone yet, right?
Definitely.
Yeah.
So, and the last question from Simon also, it's for Christian. He is asking whether you can clarify if the ontogeny links bulk to single cell level, if both are available, and how HLA and germline variation are linked. We can't hear you.
So,
oops.
Can you hear me now?
Yeah, good.
Okay. I'm not sure whether I get the question correctly. So number one,
if the ontogeny links bulk to single cell level, if both are available.
If both are available, well, let's put it this way: an experiment is never both things. It's either a single cell experiment or a bulk experiment. You can degrade a single cell experiment into a bulk experiment very easily by simply throwing away the identifiers, but then you have a very small bulk data set. So this is nothing that has anything to do with the ontology linkage. That's a question of our data structure. So then there was a second part about HLA and
how HLA and germline variation are linked. But I think this is
okay. So the HLA, this is one of the things that we are currently working on. The HLA is, so both things, HLA and germline haplotype, are properties of an individual subject in humans. So these things will be linked on the level of the subject record. And right now, the idea is that basically you have an array of information of the genes that are there. This, of course, gets tricky very quickly. For HLA, it's relatively simple because in the end, there is only a limited number of genes, but the IG and TCR loci are, of course, notoriously polymorphic. So we're trying to come up with ways how to do that in a simple way, but right now what we're working on seems to be a rather comprehensive solution, but that will take a little bit further.
Thank you.
Maybe just to add, just on the single cell and bulk. So the AIRR standard has a cell object and a sequence or a rearrangement object. So, you know, the standard has the capability to do that. We don't have a lot of data sets, or we don't really have any data sets in the AIRR Data Commons that both have cell with gene expression data and ultimately receptors with antigen specificity eventually is what the goal and the vision is. And you could certainly have, if you did a study with bulk and single cell, and they were complementing each other, you know, the standard supports all that. We just don't really have any data that reflects that. So almost all of the data in the AIRR Data Commons is bulk data right now, or single cell data, where we basically just have the B cell and T cell kind of annotations from that single cell study. So there are single cell studies, but we don't have the cell and gene expression data in the repositories yet.
All right. So I think we are getting to the end. There is no additional question in the chat. So again, I would like to thank everyone for this, the great presentation, the great talk, and the discussion. Of course, we would like to thank at the level of the AIRR community, the FOCiS committee, for providing us this opportunity to organize this first symposium. The Antibody Society, who is hosting us as a committee. I would like to thank also the AIRR meeting subcommittee, and in particular Pam Borgard, who has been dealing for many of this organization behind the scene. And at the end, all the speakers and the participants for the questions and the exchanges. I hope, and we hope, I think I can talk for everyone, that this first symposium has been interesting and providing you valuable information on how AIRR-seq data can